All right, let's start. Okay, so today we're going to talk about gluconeogenesis. You know, we have lectures back to back, gluconeogenesis, and later on the glucogenolysis. Well, gluconeogenesis is nothing else but the synthesis of a new glucose. Okay, these are the learning objectives. You can read those on your own. So that's the first case. Okay, a non-alcoholic in an apartment building was found lying down unconscious in the bottom of the staircase with a broken nose. So he was found by his neighbor. So the neighbor called the emergency department. The ambulance came pick her up and pick him up and take her to the emergency department. And he was later on analyzed. Blood was taken and analyzed. So the ionic gap was 36, and the normal range is between 9 and 15. And his blood alcohol level was 249, which is within intoxication range, which is 150 to 300. So basically, this man was intoxicated. His blood glucose was 37 milligram per deciliter, which is low normal. So the guy is hypoglycemic. Okay, so the fact that the patient has a large ionic gap and also the hypoglycemia. So what may be the explanation of this situation? What's going on? Okay, we're going to answer that. And then later on, the biochemical basis of the hypoglycemia and due to alcohol situation. Okay, so I don't know if you had a chance to answer the question, but we're going to answer as we go, okay? So remember two things. Because students tend to confuse the gluconeogenesis and the glycogenolysis. Basically, this one, the gluconeogenesis, you synthesize a new glucose, and both of them contributing to the maintaining the blood glucose. And the glucogenolysis, basically, you hydrolyzing the glycogen from your liver or in the kidney in order to maintain your blood glucose. So even though both pathways contribute to maintaining blood glucose, they are different, okay? So keep that in mind. So this one is the breakdown, and this one is the synthesis. All right, so now we're gonna talk about the gluconeogenesis. Gluconeogenesis, basically you synthesize the new glucose, as I mentioned, from non-gluconeogenic molecule, 3 to 4 carbon molecule, and in most cases, pyruvate, and sometimes lactate, okay? And it occurs in the cytosol, okay? Now, the small portions occur in the mitochondria. So keep that in mind. Only one reaction occurs in the mitochondria, but the rest take place in the cytosol, okay? So now, it turns out the substrate, in many cases, travel through the bloodstream, and it's like a reverse of the glycolysis, but not quite. Keep that in mind, okay? And we're going to differentiate them. So that basically the pyruvate link the glycolysis and the gluconeogenesis, so they're tightly regulated. So when one is occurring, the other one is shut down. If the other one is occurring, the other one is shut down. So keep that in mind, okay? They don't take place in the same time. That would be crazy, right? Synthesizing and breaking it. So you already had a glycolysis. So I don't need to go over that in detail, but I'm going to point out some irreversible steps, and then we're going to talk about gluconeogenesis and what will be the difference. So these are tissues that depend on the glucose. Really, your brains really utilize a lot of glucose, okay? When you're in a welfare state, red blood cells also use, basically they depend on glycolysis because they don't have what? Mitochondria. If you don't have mitochondria, you cannot use fatty acid for energy purpose. So kidney, medulla, and the cornea, they're also, even though they have some mitochondria, but they don't have enough, even the brain cannot utilize fatty acid, really. But they can use ketone body. In many cases, because the mitochondria, either in the case of red blood cell, they don't have mitochondria, or they're not rich in mitochondria. But the red blood cells do not have mitochondria. Keep that in mind. So it turns out during fast, again, this is what I was saying. Anytime you start talking about biochemical processes, metabolism, you have to put yourself in a condition. So either you're in a fast state or you are in the well-fed state. Keep that in mind, okay? So during fast, the glycogen breakdown occurs primarily to maintain the blood glucose, okay? Within the 24 hours, if you see here, when you are well-fed, okay, before the starvation condition, when you are well-fed, your blood glucose is maintained by the food you eat, right? So within the 4 hours, the food that you ate, the glucose level that was maintained due to the food you ate start dropping, you know. It's like at 8 o'clock you eat, and then by noon you're hungry, you need to eat, right? Because your blood glucose starts dropping. Now, once the blood glucose drops to a certain extent, and then glycogen breakdown will start taking over to try to maintain you until your next meal, okay? So once you take time, you don't eat. For some people, you know, you're in the classroom, you don't want to bother, you sit down 8 hours, you're not eating. So your glycogen storage starts getting depleted. Within the 24 hours, your glycogen storage is depleted, okay? So the body will say, no, I'm not going to wait for that. I will start synthesizing glucose from scratch so I don't have to utilize all the glycogen in the storage, okay? So the gluconeogenesis will start taking place at that point. 4 hours, so within the 24 hours when your glycogen storage is depleted, gluconeogenesis really is at peak, okay? And the gluconeogenesis then plays a big role in maintaining your blood glucose at that point, okay? Now, if you go beyond that, then fatty acid mobilization, all the stuff start taking place. We're gonna talk about those stuff, okay? So tissue that go gluconeogenesis tissues, basically liver and kidneys play a big role in maintaining your blood glucose. I'm sure you're gonna read it also, gluconeogenesis taking place in the muscle, but believe me, muscle only care about itself. So basically when the glucogen is synthesized in the muscle, it's only get utilized by muscle. I make it, I use it myself. Don't care about anybody else. That's the muscle. But liver cares about everybody, okay? So it synthesizes glucogen, save it, and you release it to the blood for everybody else to utilize it. But kidney also play a role in the gluconeogenesis, but kidney play a bigger role in, later in the prolonged fasting, when you really, the liver is overwhelmed, and the kidney start taking over, because at that time, your muscle breakdown start occurring. Amino acids from your muscles are now being traveled to different tissues, including the kidney, and the kidney start playing a big role at that point. That's in the prolonged fast, okay? Now, as I mentioned, you've seen the glycolysis and gluconeogenesis, not 100%, but it is a reverse of glycolysis. So we'll compare the two. It turns out, in many cases, many steps, it is a reverse, okay? Except three major steps that are irreversible. Those are the steps that you have here, the first, and then the PFK1 and the pyruvate kinase, because they are highly exergonic. If you recall, they're highly exergonic reactions, so they're not reversible, okay? So it's these three steps. So now, in terms of the first one, the second, and the third, now it turns out in order for them to basically reverse, let's put it that way, is completely different chemistry, completely different enzyme involved with those three steps, and we'll call those bypasses, okay? So those are the three we're gonna really emphasize on, specifically this area, because you need a completely different enzyme in order to reverse those reactions, okay? So now, these are the enzymes that are mentioned in the glycolysis. Now, these are the bypasses, okay? The bypass 1, which is right here, from pyruvate to phosphoenolpyruvate, and the bypass 2 from fructose 1,6-bisphosphate to fructose 6-phosphate, and the bypass 3 from glucose 6-phosphate to 3-glucose, and now it is released, right? All right, so we're gonna talk about those three bypasses, and these are the enzymes that catalyze the glycolysis. Now we're going to talk about the enzyme that catalyze the gluconeogenesis in those bypasses. All right. So again, this is glycolysis, glucokinase. Remember glucokinase? And what is the other one? Exokinase. What is the difference between the two? Glucokinase is found where? In the liver and in the pancreas. How about the exokinase? Very much everywhere, right? Good. All right. And then what is the difference between the two? They're both phosphorylated, but kinases. What is the difference in terms of enzyme kinetics? Different Km value. And that is very important. The glucokinase will not touch glucose, will not phosphorylate until the blood glucose reaches a certain point. And there is a reason for that. If you recall, I mentioned that. And I'm going to talk about it even more. Because when you phosphorylate, the glucose is trapped in the cell. And you don't want to trap glucose inside the liver until you're ready to lay it down as a glycogen. So keep that in mind. So that's why you have a glucokinase. And the exokinase has a lower Km, found in all the tissues. So once the glucose shows up, you phosphorylate to trap them so they can be utilized, specifically in the muscle tissues. Okay? So the bypasses, now the first bypass, in order to reverse the reaction catalyzed pyruvate kinase, you have two enzymes now, okay, in the gluconeogenesis. The first one is the pyruvate carboxylase, okay? And the second one is the PEP carboxykinase, phosphoenol-pyruvate carboxykinase, called PEPCK, okay? So two enzymes just to reverse one single reaction that is catalyzed by pyruvate kinase. We're going to talk about all of them in detail. But the second one is the fructose 1,6-bisphosphatase to reverse the reaction that is catalyzed by fructose 6-phosphate kinase. The fructose 6-phosphate kinase to fructose 1,6-bisphosphate. Let me repeat that. The PFK1, phosphofructokinase 1, catalyzes the phosphorylation, basically second phosphorylation, of the fructose 6-phosphate to fructose 1,6-bisphosphate. Is that clear? Good. So now you want to, in the gluconeogenesis, you want to reverse this reaction. The fructose 1,6-bisphosphate has to be converted now to fructose 6-phosphate. So basically, you are removing one phosphate from the position 1. Is that clear? All right. So that catalyzed by the enzyme called fructose 1,6-bisphosphatase. So the phosphatase will come and remove the phosphate from position 1 on the glucose. Now you end up to glucose 6-phosphate, right? So the glucose 6-phosphate, fructose 6-phosphate is now converted to mutate by the mutase to glucose 6-phosphate. And the glucose 6-phosphate now is the phosphate is removed by this enzyme called the glucose 6-phosphatase. You know this enzyme, right? All right, good. So then this enzyme, glucose 6-phosphatase, remove the phosphate off the position. Now you have your free glucose that is released in the blood. Now, this glucose 6-phosphatase is not present in the muscle, right? Why is that? If you recall, I said the muscle do not care about anybody else, do not contribute in maintaining the blue glucose. So even though gluconeogenesis occurs in the muscle, but it's not to provide the energy to other tissues. No, it's to synthesize its own glycogen for storage for itself. Okay, so it does not. So it doesn't have this enzyme to remove the phosphate from glucose 6-phosphate, so you have a free glucose released in the blood. Is that clear? Keep that in mind. So those are the reason I'm insisting on this, because people like in the board exams, so they like to pick up on those small differences. Okay, so the muscle do not have glucose 6-phosphatase. Okay, everything else there, but except the glucose 6-phosphatase. All right, so bypass 1. But the reason is, as I said, muscle cells do not control. Gluconeogenesis takes place in the muscle. Gluconeogenesis takes place in the muscle. But once you reach the glucose 6-phosphate, the phosphatase that remove the phosphate from glucose 6-phosphate to have free glucose is not present. And the reason for that, to maintain the glucose in a phosphorylated form in the muscle, so it doesn't get released, okay? And it stays trapped in the muscle, okay? But the kidneys and the liver, they have the phosphatase to release it. Is that clear? All right, good. So now, this is the bypass one we're gonna talk about now, okay? So here is, in a detail, now the first reaction occurs in the mitochondria. Keep that in mind, okay? And again, that's a particularity, keep that in mind. So the first reaction occurs in the mitochondria, so the pyruvate come and combine with the CO2 in the presence of ATP. So basically, the ATP is to activate the CO2, and then you have a reaction that is catalyzed by the enzyme called pyruvate carboxylase. The carboxylate, because it attaches the CO2 to the pyruvate. Now you have your oxaloacetate. You remember oxaloacetate, which is an intermediate in the Krebs cycle? So when you add the CO2 to pyruvate, right there you have your oxaloacetate. But the CO2 has to be activated in the presence of the ATP because it's kind of inert. So once you have the ATP, then that will activate the CO2 in order to interact with the pyruvate, and then you have your oxaloacetate. And so that occurs in the mitochondria. So you have a specific transporters that carry the oxaloacetate outside. Now the oxaloacetate, can act on by a second enzyme called the PepsiK, the phosphoenolpyruvate carboxykinase, if you recall, PepsiK. Now the PepsiK then converts the oxaloacetate to phosphoenolpyruvate. Now the CO2 is released, but remember, these reactions require GTP, okay? This one requires ATP, but this one requires GTP. You also require magnesium, sometimes the manganese, Mn2 plus or Mg2 plus, okay? So the pyruvate carboxylase is a very important enzyme, and it requires biotin. And this is very important. That's why I'm emphasizing on. This is very, very important. You have to have the biotin for the carboxylation to occur. The enzyme requires a cofactor. Okay, as a matter of fact, we're not talking about even a little more detail. Is this clear? So the first portion takes place in the mitochondria. The second reaction takes place in the cytosol. Okay, and everything else occurs in the cytosol at that point. Now the bypass 2. Now the bypass 2, it turns out fructose 1,6-bisphosphatase. Now, between the two, the bypass 1 and bypass 2, you remember there are glycolysis 10 steps. But the remaining of the steps that we're not going to discuss further is because they are reversible, and those basically reactions are at equilibrium. So basically, they're depending on the substrate concentration. So if the product concentration is higher, so the reaction go in directly in the opposite direction, and are very much in equilibrium. Okay. So, depending on the substrate, one way, you go the other direction. If this substrate is higher, you go in the opposite direction. So basically, they are at equilibrium. So that's why you don't need a bypass here. So very much close to the equilibrium. The concentration of substrate product, the ratio is very close to 1. Okay? Is that clear? So these reactions are reversible. But now the next one is a bypass 2 that is catalyzed 1,6-bisphosphatase. Basically, water comes hydrolysis to remove the phosphate from position 1, and you have the fructose 6-phosphate. Now you have a mutase here. Mutase basically takes the fructose 1. The fructose 6-phosphate is mutated to fructose glucose 6-phosphate. Okay. You remember, the mutase just takes this and convert it to a glucose 6-phosphate, and that reaction also is reversible. Is that clear? Now, the glucose 6-phosphate now cannot release the phosphate until the phosphatase come along, take the the phosphate off, and take it off, and free the glucose. Now that can be released. Okay? So the initial reaction to glycolysis is catalyzed by glucokinase, exokinase, but the reverse is catalyzed by the phosphatase, glucose 6-phosphatase. Is that clear? So those are the 3 bypasses. Now you have 3 enzymes in glycolysis, but you have 4 enzymes in the gluconeogenesis. in order to bypass those reactions. I'll talk about those irreversible steps, okay? Total of 10 steps in here, and here it's 11 steps. Is that clear? Okay. All right, substrate. Now, for gluconeogenesis, you have different substrate. Pyruvate come on top of the list because pyruvate can be converted directly to phosphoenolpyruvate by the two reactions we just described. But glycerol also can play a role. So the glycerol come, specifically when you have a fast for a long time or starvation occur for a prolonged period. So it turns out your fatty acids start getting mobilized. Okay, the fat breakdown occur. So the fat breakdown occurring, you remember triacyl glycerol? So glycerol backbone with 3 fatty acids attached to. So when the 3 fatty acids are removed, so now you end up with a 3 glycerol, right? And that glycerol can be carried all the way to liver and get phosphorylated. Okay, forgive me, do not try to memorize this entire thing, okay? I just put there for you to connect the two. So don't try to memorize it. It's not good for you because it's gonna take all your time for no reason. So I just want to point it out to you for you to know where each one of them are coming from. So the glycerol will come from the triacyl glycerol getting breakdown because you need the energy. Fatty acid to start to contribute now. So once that occurs, the free fatty acid that are removed from the triacyl glycerol, the glycerol backbone, can be utilized in order for gluconeogenesis. Because first of all, when you take it to the liver, it gets phosphorylated by glycerol kinase, now get converted to hydroxyacetone phosphate, which is an intermediate, and that can move on in the gluconeogenesis, so it can be synthesized. It doesn't contribute much, but at least contribute some, okay, with the prolonged gluconeogenesis. But lactate also play a role. Lactase is basically when you reduce the pyruvate, so you can oxidize the lactate back again to pyruvate, and the pyruvate can go to gluconeogenesis. Is that clear? Now, that's the amino acid now. Okay, this is an important step. In a sense, if you recall, with the heavy prolonged fasting or starvation condition, glucose is not coming, you start running out of your pyruvate. Now your body has to find a way to get the substrate. At that point, you start breaking down your muscle tissues to release the basically break down proteins in order for amino acid to be released. Those amino acids can contribute to the gluconeogenesis. Now, you have 20 amino acids. Each one of them can get into the Krebs cycle or to the gluconeogenesis cycle one way or the other. Okay, there are different portions. Again, don't try to memorize this. I want to point out what you need to know. Now, certain amino acids, alanine, cysteine, and so forth, can give rise to pyruvate. Okay? Now, others can, for instance, your aspartate, phenylalanine, and tyrosine can feed into the Krebs cycle. So once they feed in the fumarate here, they can be converted back to malate and then come out as a pyruvate through the oxaloacetate coming out of the mitochondria. Okay? And the oxaloacetate can be converted to what's called the phosphoenolpyruvate. Okay? So that's how the aspartate and the phenylalanine and others. So isoleucine, methionine, valine, and threonine can also convert it to propionyl-CoA, and they can get into your Krebs cycle and then can come out as an oxaloacetate. Now, you have others that come in, but few things that I want to bring it out to your attention. The leucine, phenylalanine, tyrosine, tryptophan, lysine, these give rise to acetoacetate, okay? And then you have others can give rise to acetyl-CoA, acetyl and acetate and acetoacetate. So those are called the ketogenic because they give rise to acetyl-CoA and acetoacetate. They're called ketogenic because once the acetoacetate is formed, basically you're in the process to synthesize your ketone bodies, including the acetyl-CoA. But we're going to emphasize that on that a little more, okay? So, and then again, it is a fine line. Because some of them can also give rise not only to be gluconeogenic, but they also can be ketogenic. Because some of them are long. When they get metabolized, they can give, some of them can give, for instance, if you look at an example, a tyrosine, for instance, tyrosine gives rise to acetoacetate, and tyrosine can also give it a fumarate. So all of those that can directly feed into the Krebs cycle are called gluconeogenic, but those that give rise to pseudoacetate or acetyl-CoA are called ketogenic. And again, there is a fine line. I'm not going to ask you to read it, but I just want you to know it, okay? Is that clear? If they give rise to acetyl-CoA or acetic, they're called ketogenic. If they give rise to something that gets directly to the Krebs cycle, they're called gluconeogenic, because the carbon skeleton can directly come out as oxalic acid and contribute to the glucose synthesis to gluconeogenesis. Is that clear? Is that clear? Good. All right. So the lactate, if you recall, the lactate is generated in your hemocytes, red blood cells, but also tissue that is exercising vigorously. The pyruvate oxidation in the Krebs cycle, due to low oxygen concentration, cannot catch up really. So the pyruvate start accumulating. That pyruvate then get converted to lactate, and lactate get taken out of the cell from your muscle cells, now get released in the blood, and it's taken down to your liver. So the lactate get converted to glucose through the gluconeogenesis. So same thing occurs in your red blood cells. They don't have a mitochondria, so glycolysis, the pyruvate is present, get converted to lactic acid, and the lactic acid now transported outside of the hemocytes to the blood, and then travel all the way to the liver where the gluconeogenesis takes place. The lactate only has to be oxidized, it becomes pyruvate right away, okay? And that the enzyme that play a role is called the lactate dehydrogenase. And this is very important, and that's why I'm emphasizing, we're gonna talk about it a little more when you talk about alcohol metabolism to a certain extent, okay? And this process is called Cori cycle. Glucose oxidized, break down all the way to pyruvate, lactic acid formation, and take it back to the liver. Gluconeogenesis occurs, glucose is reformed, go back in the blood, and so forth. So it's called the Cori cycle. Is that clear? All right. Now, if you remember, I said the glycerol backbone from the fatty acid. It's right here. The 3 fatty acids is removed. The glycerol backbone now is free to get into the gluconeogenic pathway, but it has to travel all the way to your liver, where it gets phosphorylated to glycerol phosphate and get it to the gluconeogenic pathway. So you know now where the glycerol comes from, right? All right. So amino acid, as I said, this occurs when you have a prolonged fast. If you remember, now you're still struggling for glucose, your muscles start getting breakdown for the amino acid, and that's something you don't want, right? So that's why when you're in a prolonged, you're trying to lose weight, don't starve yourself too much, get some proteins on board, stay away from sugar, which is not bad, but get on board some proteins so you don't break down your own muscle cells in order to provide amino acid for gluconeogenic pathways. Is that clear? Okay. So again, you see pyruvate can go to gluconeogenesis, and oxaloacetate can also go directly. So those amino acids that contribute directly to Krebs cycle and the carbons come out as oxaloacetate are called the gluconeogenic. Those that do not get to the Krebs cycle directly but converted to acetyl-CoA or acetate, those are called ketogenic. Is that clear? And I say there is a gray line. I'm just one of you are going to look at what Dr. Traore, you said that these are ketogenic, but they're present here. Yes, true. As I said, some can not only, it's a larger molecule, a piece can get into the TCA, but also a piece can go to the acetyl-CoA or acetate formation. Is that clear? Ketogenic and gluconeogenic. Gluconeogenic, you get to the Krebs cycle. Ketogenic, you give rise to acetyl-CoA or pseudoacetate. All right. Now, other molecules can contribute. So you know most of them now, glycerol, the alanine, the amino acid can convert to pyruvate directly. Lactic acid can convert to pyruvate. Amino acid can convert to pyruvate. But when the fatty acid breakdown occurs, okay, in general, fat do not contribute to gluconeogenic, just for energy purpose, okay, fat breakdown. They only contribute to energy purpose for gluconeogenesis. Remember, the acetyl-CoA do not get converted to pyruvate. Remember that, okay? So, because those are the questions people like to ask. Fat do not contribute to gluconeogenic pathway because the acetyl-CoA cannot be reverted back to pyruvate. But here is where fat contribute to gluconeogenesis. Sometimes if you have odd chain carbon number fatty acid, those with the ampere number of carbon. Everybody following that? When they get clear, we're going to talk about fat breakdown, maybe next week. When that fatty acid that has an ampere number of carbon, when they break down by 2, they give rise to 3 carbon at the end. So that 3 carbons molecule is called the propionyl-CoA. Okay, so 3 carbons. The propionyl-CoA now get converted to what's called methylmalonyl-CoA, that then get converted to succinyl-CoA. Now, if you know succinyl-CoA already, which is which is a product, intermediate in the Krebs cycle. Is that clear? So now the succinyl-CoA, once you get to the Krebs cycle, so the carbon skeleton can come out as oxaloacetate. So it becomes gluconeogenic at that point. So the propionyl-CoA is a gluconeogenic, but acetyl-CoA is not. Is that clear? So when fatty acid breakdown occurs, if that even number, they don't contribute to gluconeogenic. If your ampere number, then the last 3 carbons molecule, which is the propionyl-CoA, can then contribute to gluconeogenesis. Is that clear? And this process is very, very important because there are 2 vitamins involved, like B12, and then we're going to talk about that when we talk about the fatty acid breakdown. Okay? Keep that in mind. So regulation. So the regulation, acetyl-CoA play a big role in the regulation. If you remember, key enzymes for the gluconeogenesis is carboxylase. The carboxylase will convert pyruvate to oxaloacetate, is activated when acetyl-CoA level is high. So what happened, the acetyl-CoA will shut down the pyruvate dehydrogenase, which is understandable, but stimulate the pyruvate carboxylase. Why is that? What's the function of pyruvate dehydrogenase? It converts the pyruvate to acetyl-CoA. So if you already have a lot of acetyl-CoA, do you want to make more? No, then you shut this down, and then once you shut this guy down, then you stimulate so pyruvate can take another route. Basically, this occurs when you're in a starvation condition and you have a lot of fatty acid coming through the fat breakdown. Those acetyl-CoA then level goes up in the mitochondria. So you shut down the pyruvate to conserve to shut down pyruvate dehydrogenase to conserve the pyruvate, but in the same time you stimulate the gluconeogenesis at that point. Is that clear? Good. All right. So as you see here, I have a big arrow to show you which pathway. And this is very, very important. Okay. And this takes place where? In the cytosol or mitochondria? You should answer it right away. You remember the first step occurs in the mitochondria, and this enzyme is in the mitochondria. All right? Good. So this is the whole process, okay? So it is important. That's why I bring it out to you in a larger scale and a detail. So it first started with the carboxylase, pyruvate carboxylase here, which is covalently linked to biotin. If you recall, I said biotin is very important as a cofactor for this reaction. So biotin is naturally covalently linked to the carboxylase. So now it turns out the function of these biotin is the first to accept the CO2. Okay, so it's a helper for the enzyme. Hold on the CO2 now until the pyruvate come along. When the pyruvate comes, so it then passes the CO2 to the pyruvate. Now pyruvate now accept the CO2 and become what's called oxaloacetate. And the oxaloacetate is converted to malate, and malate is then taken outside and revert back to oxaloacetate, and the oxaloacetate is further converted to phosphoenolpyruvate. Is that clear? Is this clear? Pyruvate converted to malate just like in the Krebs cycle, if you recall. So basically reversing the Krebs cycle. Normally pyruvate malate is converted to oxaloacetate in the Krebs cycle. But that reaction is reversed now. So oxaloacetate to malate. So basically you have a reduction of the oxaloacetate. So malate now can be transported outside and go through this pathway and revert back to oxaloacetate and then convert it to phosphoenolpyruvate. Is this clear? Just take a look at it. It's not that complex. Here, this step is just the reverse of the small portion of the Krebs cycle. When malate get converted to oxaloacetate, now it's reversed back, and then the malate come out. And then once the malate come out, the malates get converted back to oxaloacetate, just like in the Krebs cycle. Now oxaloacetate is regenerated outside of the mitochondria. So basically, this is the mechanism to take oxaloacetate from the mitochondria outside, from the mitochondria to the cytosol. Is that clear? Good. So the regulation of the bypass 2, it looks overwhelming, but it's not that complicated. Once you sit down and read it, it starts making much more sense. You remember the bypass 2? The bypass 2, the fructose 1,6-bisphosphatase, which converted 1,6-bisphosphate to fructose 6-phosphate. And this phosphatase is activated by fructose 2,6-bisphosphate. All right. A lot of phosphate, right? All right. The 2,6-bisphosphate, I know you saw that in the glycolysis, right? What is the role of the 2,6-bisphosphate? Fructose 2,6-bisphosphate. He activated the PFK1. All right. That's through glycolysis. Now, if you want to reverse back, what would you do to the enzyme that reverses it back? You shut it down, right? So if you have a 2,6-bisphosphate, level goes up. So you stimulate the PFK1, but you shut down the what? The 1,6-bisphosphate. I'm not saying anything that is complicated. Just think about it. The PFK1 catalyzes the phosphorylation of 6-phosphate to 1,6-bisphosphate. Now, that's the reaction you want to reverse. So if the PFK1 is stimulated by one molecule, what that molecule would do to the reverse reaction? Would it stimulate it or inhibit it? You will shut it down, right? If you don't want the reaction to go in the opposite direction while you're stimulating this, you shut this one down, okay? So it will, if you have a lot of 2,6-bisphosphates around, it will shut down the 1,6-bisphosphatase. So the reaction would not go back, okay? Is that clear? Because stimulate the reverse reaction, which is the glycolysis. But AMP also telling you, okay, you're low energy, so that also will shut down the 1,6-bisphosphatase. And again, I'm saying it's not complicated. Once you read glycolysis and you understand that step, PFK1, and again, let me clear that to you. You will not get a question on glycolysis without PFK1. Because that's the rate determinant step, that's the committed step. Once the 1,6 is formed, because the fructose body has to go through glycolysis. So that step is very, very important in the glycolysis. Okay, so know that enzyme, PFK1, and know phosphatase as a reverse reaction catalyzed enzyme. Is that clear? No PFK1, no PFK1, no PFK1. And 2,6-bisphosphate, anytime you have a glycolysis, you have to know the 2,6-bisphosphate. Because that's the well-known stimulant of the glycolysis. So because it is a well-known stimulant of the glycolysis, it will oppose to gluconeogenesis. Is that clear? That should make it a little clear to you, right? All right. So the fructose 2,6-bisphosphate. I'm going to talk to you about even more detail. So that is the step here. You stimulate this guy, or you shut down this guy. Because that's the reverse reaction. Clear? Good. That's the 2,6-bisphosphate. And this molecule is very, very important. Let's talk about it a little more. Okay. The 2,6-bisphosphate is right here, which is the stimulant of glycolysis and negative regulator of gluconeogenesis, right? So the 2,6-bisphosphate is formed when the PFK2, now the PFK2, you remember PFK1 is the glycolysis? PFK2 will generate 2,6-bisphosphate. Now this enzyme is a bifunctional enzyme. It has a kinase activity, but it also has a phosphatase activity, okay? So when the phosphatase activity is inactivated, that occurs when the enzyme is dephosphorylated, okay? So when the enzyme is dephosphorylated, the kinase activity took over. Now you have a generation of 2,6-bisphosphate. Now the 2,6-bisphosphate, shut down the gluconeogenesis by stimulating the PFK1, right? So the 2,6-bisphosphate is generated from fructose 6-phosphate, okay, to form this. Now, if you want to shut this enzyme kinase activity and stimulate the phosphatase to reverse that, what would you do? What would you do? It is the kinase active when it's dephosphorylated. If you want to shut down the kinase activity, what would you do? Phosphorylate it, right? Good. All right. So when you phosphorylate this enzyme, as you see here, when you phosphorylate, the phosphorylated forms of this enzyme inactivate the kinase activity and stimulate the phosphatase activity. When that occurs, it would take the 2,6-bisphosphate and remove the phosphate off at second position. Now you have your fructose 6-phosphate. You get back to the gluconeogenesis. At that point, the PFK2 is inactivated. So how this enzyme get inactivated? By a specific kinase called the protein kinase kinase A. You know this enzyme? Protein kinase. You learned cell signaling, right? Cyclic AMP. So the cyclic AMP is generated by the action of adenine cyclase, and that occurs when the glucagon level is high. So again, let me give you a little hint on how to, I know it's overwhelming but one thing you have to remember when you study this kind of cascade, it's very long to work it out. Two things you have to remember. What is at the beginning and what is at the end, okay? So you link the glucagon to inactivation of PFK2 and activation of phosphatase, and that leads to decrease of 2,6-bisphosphate. So we can say this, when glucagon is bound, the 2,6-bisphosphate level is down. Okay, at least you can remember that right away, right? When the glucagon is up, the 2,6-bisphosphate level is down, and gluconeogenesis is stimulated. Now, how about if the glucagon is not there? The level of 2,6-bisphosphate is what? Up, right? When it goes up, what happened to gluconeogenesis? What happened to glucose? When the 2,6-bisphosphate is up, the gluconeogenesis is shut down, right? And how about glycolysis? Stimulated. Okay, if you can't remember, look at the glycolysis. Whatever stimulates glycolysis will shut down the gluconeogenesis. At least that can help you, right? So glucagon stimulates what? Gluconeogenesis. And insulin stimulates what? Glycolysis. So keep that in mind. So once you know that glucagon stimulates gluconeogenesis, if that is the case, so it will shut down glycolysis, and this is a regulator step. So it will lower the 2,6-bisphosphate level. Is that clear? At least you have something to work with now, right? So sit down and, it's not that complicated, but don't try to follow all the way. Link these two here first and to this. Forget about this area. Just link the glucagon to here, and then this enzyme, and then that'll make much more sense to you at that point. And then you can work the rest out. Is that clear? Good. And I put two together, two side by side. As I said, it is a bifunctional. As you see here, this is in absence of glucagon, and this is in the presence of glucagon, and the gluconeogenesis is shut down. if 2,6 is there, but because of the 2,6 is down, so the gluconeogenesis is stimulated. So here, the 2,6 bisphosphate is up, so gluconeogenesis is shut down, and glycolysis is stimulated. Okay? All right. So bypass 3, and this is where we'll talk about the enzyme, the phosphatase. You know these enzymes already. So that's the enzyme that is inactivated in the Van Gogh disease. Okay? So that's put together. Just, this is the energy requirement. So gluconeogenesis is an energy required process. So as you see, you have ATP utilization here, ATP here, NADH is here. So a total of 4 ATP and 2 GTP is utilized, and 2 NADH is utilized in the gluconeogenesis. So require energy. Where does energy come from? Fatty acid breakdown, okay? So we're gonna link those two, when we talk about furthermore on the regulation, okay? So the demand, the tissue that demand the brain length, RBC, cornea, kidneys, and testes, and the muscles that are exercising. But one thing you have to remember here, the brain relies solely on glucose when you have a high blood glucose. But it can use other molecules in a starvation condition. And that includes the ketone bodies, the 3-hydroxybutyrate acetate, and so forth, okay? And at that point, a little bit of amino acid, a little bit of glucose. That is in a starvation condition, okay? Again, the brain do not utilize fat directly. It's mostly glucose or the short molecules such as the ketone bodies, okay? So clinical correlate, there's a few things that I want you to remember, is that when you have, a study says that the babies, specifically, those babies that are prematurely born, the susceptibility to hypoglycemia is elevated. Basically, the question is, what's the molecular basis on those observations, okay? And this was a 10-year studies that the premature babies are susceptible to hypoglycemia. But one thing you have to remember, children in general have a susceptibility to hypoglycemia because the reason is quite simple. They have a large brain to body ratio, and the brain utilizes a lot of glucose. So because they have a larger brain body ratio, so they utilize quickly all the glucose around. So they are prone to hypoglycemia. But newborns are even more susceptible because they have not only they have a limited gluconeogenic pathway capacity due to a low expression of the PepsiK. The PepsiK, the rate determining step, but they also have a low ketogenic capacity. They can synthesize a lot of ketone body to help the glucose utilization. So if you don't have a ketone body, so your body gonna rely mainly on the glucose, so the glucose level can go down very quick. So premature baby, now even more, those babies that are born with, before term, because the reason is they have a smaller glucogen storage in their liver. So you can imagine, not only the brain utilize a lot of glucose, but they don't have enough storage. So very quick, they can run out of the glycogen storage, and they are prone to hypoglycemia very quick that way. Okay, and then the case, I hope you went over this case. What is the answer on this? And the alcoholic guy, read it earlier? All right, so basically it turns out the gluconeogenesis inhibited. If you went over, it's good. If you didn't get it right, because we have an answer here. So this is what happened when a person is alcoholic, okay? Everybody following this? This is a very one, I'm sure you're gonna see that in order, but I think it's best for you to see the biochemical cascade where the alcoholism can lead to hypoglycemia. There are other reasons, but this is one of them. So when you have a lot of ethanol on board, the ethanol is converted to acetaldehyde, and that can also be converted to CH3, which is acetic acid, catalyzed by, the first reaction is catalyzed by alcohol dehydrogenase, and the second reaction is catalyzed by aldehyde dehydrogenase. The fact that they are dehydrogenated, they generate a lot of NADHs. Okay, a lot of NADHs. Now, if you have a lot of NADHs around, okay, and you still generate a lot of acetate, The acetate can be converted to acetyl-CoA. So basically, you're going to have a lot of aldehydes around. And what happened, these a lot of NADHs around. If you recall in glycolysis, when you generate a lot of NADH, you deplete the NAD+, right? So you have to regenerate the NAD+. So what your body does in order to regenerate the NAD+ is to convert the pyruvate to lactic acid. So the alcohol metabolism generate a lot of NADHs, and those NADHs has to be reverted back to NAD+. So what they'll do is just pass the electron to pyruvate to lactic acid. So that will take all the substrates away from gluconeogenic pathway. Does that make sense? So pyruvate converted to lactic acid, and lactate level goes up sky high. So then the substrates are basically taken away from here, and that can lead to shutdown of the gluconeogenesis because the demand is too high and you're not being able to maintain the gluconeogenesis because substrates are taken in these directions. So that will shut down the gluconeogenesis, and your glucose level goes down because you cannot maintain the glucose level through gluconeogenesis. Okay? And there are other stuff, you generate a lot of acetyl-CoA too. That's why people taking too much beer at the beginning, from starting right away, start taking beer and start getting chubby because they're getting a lot of energy from the alcohol. You see, acetyl-CoA generates a lot of energy, right? Because they get oxidized. And acetyl-CoA can be utilized for fatty acid synthesis, and the person gets fat when they start drinking alcohol, especially children. When they reach 21, they start drinking a lot of beer. By the time you know, they start getting a little chubby, even though they don't eat much, because alcohol generates a lot of acetyl-CoA. And acetyl-CoA is the building block for fatty acids. So you start laying fat a little bit, and you start getting a little chubby. Is that clear? All right, any questions? You're almost there? If not, thank you. We have another class. And the next lecture within 10 minutes, okay?